A new variant, as shown in this figure, a series of tiny mutations found in many British samples of the coronavirus may help the virus spread more easily. The coronavirus variant is known as B117. As shown in this figure, what happened when the new variant spread? Over the last few months, the United Kingdom has faced a rapid increase in COVID-19 cases in South East England. On December 14, UK Health Secretary Matt Hancock first announced that a variant called B117, analysis of a viral genome sequence data identified a new variant which is identified by multiple spike protein mutation. One mutation called N510Y makes this virus bind more tightly to human cells, perhaps by helping it bind better to ACE. ACE, a host protein that lets virus into cells. This mutation has also appeared independently in a rapid spreading variant in South Africa. A preliminary analysis in UK suggests that the variant is significantly more transmissible than previously circulating variant, with estimation potential increase of predicted number by 0.4, or greater, with an estimated increased transmissibility up to 70%. Is the SARS-CoV-2 virus unusual? The average mutation rate SARS-CoV-2 remains low and steady. It's very important. And it is much slower than other RNA viruses, such as influenza virus. Unlike coronavirus, influenza virus, which is caused by flu, are prone to change through a process called antigenic drift and antigenic shift. As this figure, antigenic drift refer to accumulation of a genetic mutation that causes an alteration on the surface of the virus. The mutated antigen drifts. So, from original conformation. This is one of the main reasons why novel flu vaccine is required every year. So, antigenic shift occurs when segments from the genome of the two different viruses make a novel strain. Coronavirus are not prone to undergo antigenic drift or shift. Sometimes multiple mutations occur quickly, as was the case with B117 variant in UK. It is estimated that B117 is about 30 to 35 mutations away from the original strain detected in Wuhan, China at the beginning of the pandemic. The more spike protein change, the harder it is for the immune system of the people who have been vaccinated to mount a swift attack. The same goes for people who have already had COVID-19, their immune system numbed the spike protein of the older variants. As shown in this figure, currently, as you see, there are 3 variants of concern, which are named based on the country where they were first identified. The UK B117, as shown in this figure. The South African B1-351 and Brazilian P1 variants have a mutation which are thought to make them more transmissible. There are also early data to suggest that UK variant is associated with a higher mortality risk than the white type SARS-CoV-2. The mutation in these variants, led to a small change in the structure of spike protein, which is the protein used by the virus to enter a human cell. The spike protein also part of structure of the virus where utilizing the antibody bind to block the SARS-CoV-2 infection. Consequently, this mutation can affect SARS-CoV-2 in any advantageous or disadvantageous way. So, what does 614G mean, as in this figure? Let us explain what meaning 614G. Or D614 means the original form, the mutant form referred to as D614G or 614. The coronavirus that causes COVID-19 uses it is a spike protein to infect a human cell. So D614G referred to an amino acid mutation. This is a protein that has become increasingly common in SARS-CoV-2 virus from around the world. The spike protein S is a string of 1,273 amino acids. In the original form from Wuhan, the 614 of these amino acids has the chemical symbol D as aspartic acid, as shown in this figure. While in mutated form, the 614 amino acid abbreviated G-glycine, or so D614G is short for having spike protein with aspartic acid at position 614 mutated to glycine. As shown in this figure, changes in a global distribution of relative frequencies of the D614 orange color and G614 G blue color variants. Circle size indicate the relative sample within each map. So as in this figure, there is some preliminary evidence that's more contagious. I mean talk about the G614, the new variants. So, more contagious. Scientists noticed a surge of cases in areas where the new strain appeared. Possible implications for human health. Most mutations that emerge will not provide selective advantage to the virus. However, some mutation or a combination of mutation may prove the virus with a selective advantage, such as increased transmissibility through an increase in receptor binding or ability to evade the host immune response by altering the surface structure recognized by antibodies. So the previous investigation of the D614G variants identified that while the 614 variant provide a selective advantage, so increased cellular infectivity, there was no identifiable effect on infection severity or outcome. As shown in this figure, potential impact on SARS-CoV-2 diagnosis. The UK report that the deletion, the spike protein of the variants cause negative result from the S gene. Real-time PCR assays applied in some laboratories in UK. So this is specific mutation has occurred many times in different countries and geographically widespread. Assays targeting the S gene are not widely used for primary. So detection and only one assays targeting the S gene is one of list published in house assay list by WHO, relying only on S gene for primary detection. SARS-CoV-2 infection using real-time PCR is not recommended because mutations are more likely to occur in this gene. So, Implication of the emerging SARS-CoV-2 variant on efficacy of the COVID-19 vaccine. This is one way to assess whether the current vaccine work against emerging variant to expose blood sample from the individual who have been vaccinated against or previously contracted COVID-19 to new variant in vitro. So, in doing so, scientists can determine the relative ability of the participant antibodies to neutralize the novel variant compared with the original or wild type virus. So, strategy to overcome vaccine escape. As in this shown, this is a slide, strategy to overcome vaccine escape. Firstly, we must know what mean by vaccine escape. So vaccine escape is the term used to describe the process of a virus mutating to form a variant that evades the immune response induced by vaccination. This is a process is rare, but occurrence depends on the factors such as therapeutic target and the viral mutation rate. An explanation above, as I mentioned, in the case of SARS-CoV-2, the mutation rate is low, okay? So however, the virus is so widespread, numerous variants have developed novel variants may be reduced capacity of antibody to neutralize the virus in individuals who have previously contracted or had vaccination against SARS-CoV-2. In fact, decrease in the neutralization power of previously developed antibodies against the South African variant has been consistently observed. Nevertheless, vaccine allow to a multitude of different antibodies and T cells to be produced against a numerous part of spike protein, and such as it is expected that is a certain level of protection should still be maintained. To ensure maximum level of protection, strategies have been devised to overcome a decrease in vaccine efficacy. So, strategy to increase vaccine efficacy. The vaccine administration can be modified to increase the overall immune response and ideally provide more protection against new variants. Example, additional booster vaccine does not be considered. Optimization of the original vaccine, such as the development of a new version with updated spike protein is also possible. However, while the direct change to the vaccine can be performed relatively quickly, further steps to be required ensure quality, safety, and perhaps effectiveness of a new vaccines. So, now the question is a very important question. Shall we give the vaccine, although we expect that the new SARS-CoV-2 variant will emerge over time? Experts agree that it's essential that a current vaccine continue to be administered to as many people as possible. This is because of the protection offered by the vaccine is greater than the risk of the vaccine escape from the potential new variants. New trend, what about the new trends? The evidence suggests that the available COVID-19 vaccine may still produce protection immune response against the new variant identified to date. However, the level of efficacy, especially against severe disease, is yet to be defined. Changes to the vaccine can be made to the target novel variant and will likely be needed to as a new variant will become dominant among the virus that is circulating. So, finally, rigorous and increased compliance with public health mitigation strategies such as vaccination, public or physical distancing, use of mask, hands, hygiene, and isolation. Quarantine is essential to limit the spread of virus that cause COVID-19 and protect public health. Thank you so much for listening.